I want you to picture something for me. When you hear the words Alzheimer's disease, what is the very first image that pops into your head?
I mean, for most people, it's definitely amyloid plaques.
Right, exactly. If you're like most listeners or even most scientists, you're picturing those dense clumps of toxic proteins. We've been taught for decades that they are the ultimate villains, just slowly suffocating the brain's delicate networks.
But what if we've been looking at the wrong part of the crime scene this entire time?
Yeah. What if the plaques themselves aren't actually the direct cause of the memory loss and the cognitive decline? So that is our mission for this deep dive. We are exploring a truly groundbreaking paper published in Nature Aging that completely decodes these microscopic, catastrophic traffic jams that form around the plaques.
Right, and these pileups are called plaque-associated axonal spheroids. And honestly, once you understand how they work, you will never look at neurodegeneration the same way again.
It's wild.
It really is a massive paradigm shift.
Yeah.
Because this research doesn't just, map out the architecture of a microscopic problem. By zooming into the subcellular level, they found a mechanism that is actually reversible.
Wait, really? Reversible?
Yes. They've proven in live human neuron models that we might be able to heal the brain and restore its electrical function without even needing to remove the amyloid plaques themselves.
A completely different way to look at the brain. So let's unpack this and start with the biology of this traffic jam. You mentioned plaque-associated axonal spheroids, which is a mouthful, so let's break down exactly what that is for you listening.
Sure.
To give you an analogy, imagine a neural circuit is a highly efficient, high-speed highway. Information is constantly zipping along it. Historically, the amyloid plaque has been viewed as this giant roadblock sitting right across the lanes.
Right, but based on this paper, the plaque is actually more like a giant boulder sitting quietly on the grassy shoulder next to the road.
Exactly. The spheroid, however, that is a massive, swollen pileup of cars right on the highway itself, completely blocking all the traffic.
That is a perfect way to visualize it. When we look at Alzheimer's brain tissue, we see that hundreds of axons, which are those long, highway-like cables transmitting electrical signals away from the neuron cell body, they just swell up.
Like balloons, basically.
Right, into these massive balloon-like structures right next to a single amyloid plaque. And interestingly, this swelling happens specifically in the axons, not in the dendrites.
And the dendrites are the branches that receive signals, right?
Yeah, so because these axons swell up so drastically, they physically disrupt the electrical conduction. They impair the entire neural network. Yeah. And we know this matters because the severity of these spheroids correlates directly with the severity of Alzheimer's symptoms in human patients.
Right. But what is actually causing them to swell up like that? I mean, it's not just empty space inside them, is it?
Far from it. If you look inside one of these spheroids, it is packed solid with enlarged defective cellular machinery. Specifically, it's full of endolysosomal vesicles and alophagosomes.
Okay, and normally those are the cell's waste management and recycling centers, right?
Yeah, they gather up cellular trash and break it down. But inside these swollen axons, they just accumulate. They completely lose the enzymes they need to actually break down the waste, so they just enlarge and sit there.
Just physically churking the inside of the axon.
Exactly.
Okay, I have to challenge this because something isn't adding up here. If there's a massive pileup of, broken garbage trucks right in the middle of a vital neural highway, why doesn't the brain's immune system just clear them out?
That's a very logical question.
I mean, you'd think the brain's cleanup crew, the glial cells, would spot this giant swollen mess, call in the tow trucks, and clear the road.
And the answer comes down to the difference between a sudden acute injury and a slow chronic condition like Alzheimer's. In an acute neural injury, let's say a traumatic brain injury, the damaged axon undergoes rapid, violent changes in its membrane tension.
Makes sense.
When that happens, it exposes a specific lipid on its outer surface called phosphatidylserine.
Phosphatidylserine.
Right. And that lipid acts as a biological eat-me signal. The glial cells recognize that signal, swoop in, and essentially devour the damaged axon to clear the way for new growth.
Like putting a giant glowing flare on a wrecked car?
Precisely. But in Alzheimer's, for reasons we are just beginning to fully understand, these spheroids persist for incredibly long intervals, sometimes years.
Wow,
Yeah,
yeesh.
without any significant glial clearance. The eat me signal either isn't being sent or is just not being recognized by the immune cells. The brain is essentially trying to isolate the damage by letting it sit there unresolved.
Which is terrifying when you think about it. It makes you ask yourself, has the brain's attempt to quarantine the damage actually become the very thing causing the memory loss?
It really does reframe the whole disease.
So just to summarize for you listening before we move on to how they studied this, the plaque isn't the primary thing blocking the signal. It's this localized balloon-like swelling of the axon around the plaque, filled with broken cellular recycling centers. And the brain's immune system is just completely ignoring it. Have you ever considered that the brain's attempt to isolate damage might actually be the thing causing the memory loss?
And because these swollen spheroids are physically tangled up in the dense, sticky amyloid plaques, they have historically been nearly impossible to study biochemically.
Right.
You couldn't just slice them out of a brain to figure out what was inside them without getting a bunch of plaque and other cellular garbage mixed into your sample.
Which brings us to our first 5-minute checkpoint. Let's do a quick, punchy bullet recap number 1 for everyone.
Sounds good.
Bullet 1. Alzheimer's memory loss isn't just about the plaque boulder. It's about the microscopic PAS traffic jams blocking the neural highway.
Right. And bullet 2. These jams are full of defective cellular recycling centers that the brain's immune system fails to clear.
Perfect. So that leads us right into section 2, because if you can't manually cut out this microscopic traffic jam, how on earth do you isolate what's inside it?
This is where they used an incredibly clever technique called subcellular proximity labeling.
Yes, the paintball technique. Think back to our crashed car analogy. Imagine you're trying to find out exactly what went wrong inside one specific car, but that car is buried at the bottom of a chaotic pileup of hundreds of vehicles.
You can't just scoop up the whole mess.
Exactly. You need a highly specific paintball that detonates and splatters fluorescent paint only on the parts inside that one specific car you care about.
And the spatial precision required to do that inside a human cell is just staggering. But the researchers solved it brilliantly. They used a specific protein as their bait.
What was it?
It's called phospholipase D3, or PLD3.
PLD3.
Right. It's an endolysosomal protein that happens to be highly abundant inside these specific spheroids. It's uniquely expressed in neurons, and crucially, it is completely absent in the glial cells, the cleanup crew.
So PLD3 is the passenger we know for a fact is sitting inside the crashed car.
Exactly. So they took postmortem brain tissue from Alzheimer's patients and applied a primary antibody that specifically hunts down and binds to PLD3.
Okay.
Then they applied a secondary antibody that binds to the first one. But this secondary antibody is special. It's carrying a payload.
The paintball itself.
Yeah, it's conjugated with an enzyme called horseradish peroxidase, or HRP. So now you have this HRP enzyme physically anchored right next to PLD3, deep inside the spheroid.
And here is where the paintball explodes.
Yes. They flood the tissue with hydrogen peroxide, or H2O2, and a special chemical tag called biotin XX tyramide.
That's a lot of chemistry, but basically...
Basically, the HRP enzyme uses the hydrogen peroxide to trigger a tiny, highly localized chemical explosion, a peroxidation reaction.
Oh, wow.
It practically biotinulates, or tags, only the proteins within the immediate subcellular vicinity of the spheroid.
But wait, the brain tissue they're using is postmortem. It's been chemically fixed and preserved so it doesn't decay. Doesn't that mean all the proteins in the tissue are basically cross-linked together like a giant impenetrable fishing net?
They are.
So how do they actually pull the tagged proteins out of that mess?
That was a massive methodological hurdle. They had to completely optimize the protein lysis extraction. They used incredibly harsh detergents, specifically a 2% SDS solution in a basic tris HCl buffer at a pH of 8.0.
So they literally melted the chemical net away.
Effectively, yes. By uncross-linking the fixed postmortem tissue, they could extract all those biotin-tagged proteins. And just to be absolutely certain they only hit their target, they validated the spatial precision with STED super-resolution imaging.
Okay, so the tagging mechanism works beautifully. But to know if this is actually what's driving memory loss in the real world, you can't just look at a random cell in a petri dish.
Right.
You need to look at the brains of people who actually suffered from the disease. So where did the human samples come from?
They utilized clinical cohorts, specifically tissue from 39 individuals from the Yale Alzheimer's Research Center and Banner Sun Health.
39, okay.
And they specifically selected 6 severe Alzheimer's cases with the absolute highest amyloid plaque burdens to ensure a massive density of these spheroids.
But when you do an extraction like that, you're going to pull out a massive, messy list of proteins. How do they know they aren't just tagging random cellular dirt?
Well, they ran the data through a brutal 3-step statistical pipeline.
Let's hear it.
Step 1. They compared the PLD3 labeled samples to control samples with no antibody, keeping only proteins with a p-value under 0.05, a false discovery rate under 0.1, and a fold change greater than 1.5.
Filtering out the random background noise. Got it. What's step 2?
Step 2. They aggressively deleted any proteins known to belong specifically to glial cells.
Ensuring they were only looking at the neurons.
Exactly. And step 3. They compared the results to unaffected healthy brains, removing any proteins with a fold change of less than 0.67. This completely eliminated proteins that are just part of normal neuronal soma or neuropil function.
It just stripped away everything until only the absolute culprits were left.
And the result was stunning. Exactly 821 proteins were definitively identified in human PAs, with all of them showing a fold change greater than 1.95.
Oh, wow. Over 800.
Yeah, and just to prove it wasn't a fluke, they found 856 nearly identical proteins in the 5xFAD Alzheimer's mouse model.
That is just an incredible feat of bioengineering. So if you could tag and map any microscopic structure in the human body, what mystery would you want to solve first? It really makes you think.
It really does.
Let's hit our second 5-minute checkpoint with bullet recap number 2.
Bullet 1. They used a proximity labeling paintball technique anchored to the PLD3 protein to tag everything inside the spheroid.
Right, and bullet 2. After a rigorous 3-step statistical filter on human brains, they isolated exactly 821 target proteins.
Exactly.
So the obvious next question is, what exactly did those 821 tagged proteins reveal? When we look inside the spheroid, what is actually causing the traffic jam?
When we analyze those proteins, they reveal 3 core biological culprits. It's a total systemic failure.
Okay, I love a good mnemonic framework. For you listening, let's call this the TCL triad. T for traffic, which is the lipids, C for cytoskeleton, which is the scaffolding, and L for lysosomes, which is the garbage.
Perfect.
TCL.
Let's start with the garbage. The L.
Right, the lysosomes. This represents a massive breakdown in proteolysis, the cell's ability to clear waste. The pathway enrichment showed massive issues with endocytosis, phagosomes, and ubiquitin-mediated proteolysis.
Basically, the trash is piling up.
Exactly. Specific proteins like cathepsin B and RAGC were heavily detected. The cell knows there's a problem, but the machinery is totally stalled. The garbage trucks are full, but the engines are dead. Okay, what about the scaffolding? The C cytoskeleton.
This is where the environment becomes incredibly chaotic. An axon relies on a complex, shifting internal skeleton to maintain its shape. The data showed massive scaffolding dysregulation.
Warping the structural integrity.
Right. Actin cytoskeletal signaling and the RRP-WAS-ping complex, which build the scaffolding, are highly activated.
Okay.
But simultaneously, RHO-GDI signaling, which normally regulates the Rho family GT passes to control that building process, is inhibited.
So it's like a rogue construction crew endlessly building steel scaffolding in the wrong directions across the highway, trapping all the cargo.
That's exactly what's happening. Active, ongoing reorganization gone wrong.
Which brings us to T traffic, and here we're specifically talking about lipid transport and metabolism, right?
Fascinatingly, yes. The massive accumulation of all these vesicles requires huge amounts of lipids. The analysis highlighted crucial proteins like APOE.
Wait, APOE, like the major Alzheimer's risk gene.
Yes, exactly. It acts as a lipid carrier. They also found high levels of TM30A, ATP8A1, and ATP8A2.
What do those do?
They form the P4 ATPase complex to regulate membrane stability. And uniquely, they found complement C3 showed the highest expression of all, which is heavily involved in both lipid metabolism and the immune complement system.
Wait, I have to stop you. Because if the garbage trucks are full and the scaffolding is completely blocking the road, why on earth is the cell continuing to send delivery trucks full of lipids to the crash site?
That is the tragic irony here.
Why wouldn't it just shut down lipid transport?
Because the axon is swelling outward, the cell detects the membrane tension is stretching. It thinks it's structural damage that needs to be patched, so it frantically signals for more lipids to reinforce the expanding membrane.
It is literally feeding the traffic jam.
Desperately trying to manage the swelling, yes.
Wow. Okay, so synthesizing this for you listening. It's a highway where the delivery trucks The lipids are endlessly arriving, the scaffolding, the cytoskeleton, is being built in the wrong directions, and the garbage trucks, the lysosomes, are totally full but have nowhere to dump their load.
The TCL triad in action.
Which makes me wonder, and I pose this to you listening, which of these three systemic failures do you think is the hardest for a single cell to recover from?
It's a tough call. They compound each other.
Time for our final 5-minute checkpoint. Bullet recap number 3.
Bullet 1. The 821 proteins reveal the TCL dysfunctions: traffic, cytoskeleton, and lysosomes.
And bullet 2. The cell makes it worse by constantly pumping in lipids to fix the stretching membrane.
Right.
But here is my biggest lingering question. Everything we've talked about so far is based on post-mortem brains. They are dead.
Yes.
How do we know how these form in real time? If you want to fix a traffic jam, you can't just look at a photograph of a crash site taken long after the accident.
You're absolutely right. To understand the real-time dynamics, they had to create a live case model. They developed a human-induced pluripotent stem cell, or iPSC, Alzheimer's model.
Oh, wow. So they grew human neurons from stem cells.
Exactly. They used NGN2-induced glutamatergic neurons, co-cultured them with astrocytes, and then treated them with oligomeric A-beta.
Epoxic amyloid proteins.
Right. And what they saw was that the spheroids actively grew within days.
Days? That fast?
Yes. And notably, they only formed around compact theoflavin S-positive plaques, not just the diffuse amyloid floating around.
So the dense boulders cause the pileups.
Precisely. And to prove the functional block, they used GCAMP-APA for neuronal calcium imaging.
Wait, calcium imaging, so they could actually see the electrical signals?
Yes, because calcium rushes in when a neuron fires. And they prove that the calcium decay time is much slower inside the spheroids.
Visually proving the electrical conduction block.
Exactly. You can literally watch the signal get bogged down.
So we have the visual proof. But earlier you mentioned a master switch, a way to reverse this. And this is where the proteomics data gets really exciting, because one specific signaling pathway was highly activated, right?
The PI3-KT-mTOR signaling pathway.
mTOR. Why is mTOR the master switch here?
Because mTOR is the master regulator of protein turnover, lipid metabolism, and the cytoskeleton.
The TCL triad. It regulates all three.
Exactly.
So can we target it? Can we just flip the switch?
They did. They treated the 3-month-old iPSC cultures with a drug called taurin-1.
Taurin-1.
It's an inhibitor of both mTORC1 and mTORC2. And it successfully suppressed mTOR's downstream effectors, specifically P70S6K and P4EBP1.
And the result? Did it clear the jam?
The spheroids shrank significantly, and when they ran the calcium imaging again, the action potential conduction improved. The signal was making it through.
The pathology is reversible.
Yes, even with the dense amyloid plaque still sitting right next to it.
That is phenomenal. It begs the question. If a master regulator like mTOR controls so many functions, what are the broader implications of targeting it therapeutically?
Yeah.
I mean, you can't just turn off mTOR everywhere without side effects, right?
True. It will require careful clinical optimization.
Yeah.
But the proof of concept is monumental.
It really is. So to bring this all together for you listening, this deep dive fundamentally shifts our view of Alzheimer's. It's not just about amyloid plaques being toxic waste that we have to demolish.
Right.
It's about the localized subcellular traffic jams they cause, governed by lipid transport, cytoskeletal warping, and lysosomal failure. And most importantly, by turning down the mTOR pathway, the traffic jam can be cleared.
We have an entirely new, viable therapeutic target.
And that leaves us with a powerful closing thought to mull over. If we can shrink the spheroids and restore electrical signals without removing a single amyloid plaque, does this mean the future of Alzheimer's treatment isn't a cure that perfectly clears the brain, but rather a bypass that just teaches the brain to safely route traffic around the damage?
It's a completely different paradigm.
It really is. We would absolutely love to hear your thoughts on this paradigm shift. Thank you for joining us on this deep dive into the microscopic traffic jams of the mind. Keep questioning what you know.